Microencapsulation, right? So we here the maximum and minimum effective concentration and therapeutic window, right? Our drug have ideal property that is remain in this therapeutic window, that is in between the maximum safe concentration and minimum effective concentration, right? So first of all we are discuss the definition of microencapsulation. So it is the process of applying relatively the thin coating polymer to a small particles of solid, droplet of liquid, and dispersion. So here only not a solid, but we are also take the liquid droplet and dispersion, right? So here that coating is done by polymer. Coating is done by polymer. These polymer are inert, do not readily biodegradable, and have a good tissue biocompatibility. These polymer have inert in nature, do not readily biodegradable, and good tissue biocompatibility. The drug release mechanism across the membrane is surrounded by the diffusion. Here the drug release mechanism is done by diffusion because here the coating by the polymer. That's why as the time is going on, that polymer are become a swell and the drug is diffused out from the inner side to outer side. Mostly the polymer is ethyl cellulose and polyvinyl acetate. Polyvinyl acetate. So here the particle are migrate. That's why it is called as a diffusion. The advantage of microencapsulation, the first advantage is masking of taste or odor, masking of unacceptable taste or odor of the drug. So mostly the bitter taste drug, if the drug have a bitter taste, then it is taste is masked by using the microencapsulation techniques. The liquid can be converted into a free flowing solid powder. So sometimes the liquid that have a stability problem or the transportation problem, then it is converted into the solid. So that is increase the stability or easy for the transportation purpose. The stabilization of the drug sensitive to the oxygen, moisture, or light. If the drug have stability problem by the external factor, that is oxygen, moisture, or light, then it is stabilized by using the encapsulation technique, or the polymer are coated on that drug, and we prevent the drug from the external environment. The prevention of vaporization of the volatile drug. If the drug is volatile and it is coated with the polymer to prevent the vaporization. Right? The reduction of toxicity and reduce the gastrointestinal irritation. Sometimes if the drug create the problem in gastrointestinal tract or they create the toxicity, then it is prevented by converting that dosage form into the microencapsulation, right? Or the in case of gastrointestinal, if they create the acidity in stomach, then we are uh, convert or polymer are coated on that drug and convert into the encapsulation. So they are not create the acidity in the stomach. So sometimes it is used for the stability purpose also. Alteration in the site of absorption. Sometimes if drug is not suitable to absorb in the stomach, then it is coated with the particular polymer. For example, phthalate, and that coated particle that is called as a encapsulation, that is pass out from stomach to the intestine, and in the basic media it is dissolved, and that drug give the maximum viability. So at that time we are alter the Here alter the absorption site. The drug release controlled by the adjusting the thickness and the permeability of the coat. So according to our formulation need, we are adjust the thickness. So sometimes we are use the lesser thickness, sometimes we are use the more thickness. So according to our need, we are use the thickness of layer. The coated particle that is compressed into the tablet and placed into the capsule. That coated particle we are uh, fill in the capsule as well as it is converted in the tablet. The zero order delivery is possible. The release rate vary with the polymer type. Mostly it is try to follow the zero order, but sometimes the release pattern is depending on the polymer type. Disadvantage of the microencapsulation, mostly the system must not be physically removed from the implant. So the physical removal from the implant site is required, right? But here the whatever we are use the polymer that is non biodegradable, rapidly not biodegradable. So that's why the physically remove that polymer from the site is uh, our one drawback. The difficulty to deliver the high molecular weight compound. So if the particle have a high molecular weight and we are again coated that particle by a polymer, then size of that particle is increasing. So when we prepare any tablets or capsule, we require uh, more volume of the particles. So it is impossible to swallow that dosage form if the size is too much. Increase the cost per unit dose. So here we are use the uh, particular equipment for preparation of the product, as well as here we are use the different volatile solvent. So as compared to conventional or as compared to tablets, here that cost is increased. Obviously, here the cost of that product is increased. Potentially, the toxicity if system fail. Sometimes the coating is not done proper, then immediately the drug is released out. It is called as a burst effect. Immediately, that all the drug are released out from the device. That's why the concentration is cross the boundary of maximum safe concentration and goes to the toxic level. So it is create a problem in patient, right? Incomplete or discontinuous coating. Incomplete or discontinuous coating. So here the coating that is not done properly, then they create the problem. That is the drawback. The clumping of microencapsulation. Sometimes two or more granules they are made a clump, and on that the polymer are coated. So when that polymer are dissolved, two or three particles immediately they are released out, and that increase the concentration at the site particular sites. So drug release is too much, not according to our calculation. So that is the problem, right? Clumping is the problem. Okay, here the microencapsulation, the drug release is followed by fixed first law of diffusion. Describe the diffusion process. Here the equation is J is equal to minus D dC by dt. Here the capital D is the diffusion coefficient in the area per time, and dC by dt is the change of concentration C with the distance x. And here the minus symbol indicate the as the time is going on, the concentration of particular device is decreased. A method of microencapsulation, generally here the two type of classification we are discussing, but mostly the microencapsulation are prepared by this that general method, right? In general classification, the air suspension, coagulation phase separation, spray drying, congealing, pan coating, vacuum deposition, polymerization, multi-orifice centrifugal process, right? Other that microencapsulation are in other way it is classified according to the physical method of microencapsulation and chemical method of microencapsulation. In case of physical method of microencapsulation, it is again divided into different types, that is spray drying, spray chilling, rotary disc atomization, fluid bed coating, stationary nozzle coextraction, centrifugal head coextraction, submerged nozzle coextraction, and pan coating. In case of chemical method of encapsulation, that is again divided into different type, that is phase separation, solvent evaporation, solvent extraction, interfacial polymerization, simple and complex preservation, in situ polymerization, liposome technology, and nano encapsulation. Air suspension, the basic principle is to disperse the solid core material in a supporting air stream and the spray coating material on an air suspended particle. Right here, mostly the drug is remain as a suspended, and from the bottom side or from the top side, the coating material are sprayed on a air suspended particle. Right. The method is the particles are suspended on the upward moving airstream as each pass through the coating zone. This core receive an increment of the coating material. So as the material is passed from one to another side, they receive the material on the coating surface and the material coated on the solid particle or on a liquid particle. The cyclic process is repeated several hundred times depending upon the coating thickness desired. So here that process is several hundred times it is repeated depending upon the coating thickness desired, right? So till that we are continue that process. After that, if we want to coat that desired, we achieve the desired thickness, then we stop the process and we obtain our product. The supporting airstream serve to dry the product while it is being encapsulated. So here we are also use the dry air. That is the purpose of that air is to dry the product, to dry the product. So these are the air suspended equipment, right? So in that the coating chamber, then partial partition column, the column support, air distribution plate, and spray nozzle is the main part of that equipment, right? The factor which affect the encapsulation process, the coating material concentration, coating material application rate, volume of air required to support the core material, amount of coating material required, inlet and outlet operating temperature, density, surface area, melting point, solubility, friability, volatility, crystallinity, and flowability of core material. These are the factors which affect during the encapsulation process. So all that factors are maintained in a critical way, right?